In this section, we shall be mainly discussing issues related to sampling. But nevertheless, before we get there, it's important that we first talk about specifications. Specifications represent that body around which all activities in the QC department revolve. So it's very important that the buyer and the vendor clearly understand the specifications and are in agreement with those specifications. Next, there should be clear understanding and agreement of how samples will be drawn, how samples will be prepared, and a very clear understanding about the course that leads to either acceptance of a lot or rejection of that lot. Now, when we receive material, how do we check for its quality? Only by testing. Now, when it comes to testing of raw materials, you have three possible strategies. You have no testing as an option, or 100% testing as another option, or acceptance sampling as the third option. Now first let's look at this case where there is no testing. Now when do you go for this? There are some special circumstances. First, that the test procedures are such that you do not have the facilities for carrying out that test. For example, checking the D value of biological indicators. Now when you don't have the facility either in your own organization or anywhere in the neighborhood where you can outsource it, then you go purely by due diligence on vendor qualification, go for the best in class and give that as a justification that everybody else also buys from the same source. He has such a great reputation in the market and therefore you're going purely by his certificate of suitability. The second option is second scenario is when the material is highly poisonous or toxic and you do not want to unnecessarily expose your people to the test process and place them at risk. Under those circumstances, regulators permit you to qualify the best possible sources for this particular material, and when the tests have been carried out at the supplier's end, you go purely by the certificate of suitability issued by that supplier. Third situation is when the material is manufactured by your own company. Now, if you have manufactured it at one plant and care is taken to see that there is no mix-up that ever takes place, and with due diligence you are able to transport it to the other unit, then the receiving unit need not repeat the tests. Now, let's have a situation where there is one trading company and two manufacturing companies all under the same management. Now, the trading company has bought the material, it has its own laboratory, carried out its own tests. Now, after the tests are carried out, part of the material is released to Unit 1 and part of the material is released to Unit 2. Now, and if care is taken to see that no mix-up takes place, then the receiving units need not repeat those tests. Now, 100% testing may not be always possible. If the test is destructive, then to that extent, this is ruled out. Alternatively, you like to reduce the number of tests. Now 100% testing would take too much time. So you go for what is called acceptance sampling. Now the acceptance sampling will give you meaningful results and inferences drawn from those results would be applicable if the sample is representative. If the sample is non-representative, then your data that you get is meaningless, can even be misleading. So I'll try to give you an example. Now let's say you get material from a particular supplier and there is variability in the process and therefore you will find that the distribution is normal as shown here. Let me caution that every manufacturing process does not necessarily produce a normal distribution. It could be a Poisson distribution, it could be a gamma distribution, it could be any other distribution. But the beautiful thing about distributions is that if you take enough and more samples, and take the average value and plot only the average value that you have got from your various samplings, then the plot of the averages invariably is a normal distribution. This is called in statistics the central mean tendency. So now let us say you have this normal distribution, you have a tail on one side and you have a tail on the other side. And let us say the specifications are two-sided. There is a lower specification limit and there's an upper specification limit. Now the two specification lines are like railway lines. They are parallel, they don't meet. And you have this distribution with a left tail and a right tail. Now as you can see over here, you have a distribution, and the sample, if it is drawn from the head, below the head of the distribution, between the tracks, then that particular sample passes the test and the batch is released. Now, if the same batch were to be tested again, or the next batch arrives in which there has been absolutely no change in the manufacturing process, but the person who is drawing the sample draws the sample from below the tail but outside the rail. Because it is now outside the rail, it is out of specifications. But it is under the tail, it is the same distribution. So the analyst has drawn a sample and this time he rejects the batch. He rejects a batch unfairly. This batch is no different from the earlier batch, or it could be the same batch where he has drawn samples twice, and once he passes it and once he fails it. This failure of a good batch, rejection of a good batch, is called type 1 error or alpha error. Now let's take another scenario. You have a certain distribution that has been in place, the process has drifted, the process has altered, and it has shifted. And it has shifted towards one side of the rail, away from the other rail. But the sample has been taken such that the sample falls between the rails. Then the analyst passes that batch. This batch is a rotten batch that should have been failed. But because the sample was drawn from a location where the results indicated that the process was in a state of control, so the analyst passed it. This is an undetected OOS that has been passed. This is called a type 2 error. This is called a beta error. In the first case, the alpha error, it is the producer's risk because the material is being returned to the producer for no fault of his. And in this second case, it is the consumer risk because the consumer is being given something that he shouldn't be given in the first place. It is a totally unacceptable batch for his process, but it has passed the test. Had, of course, the sample come from the other side, way outside the rails, but under the distribution, the analyst would have failed it anyway. But the sample he drew happened to be from the left side, and there you find that the sample happens to fall between the tracks, and therefore it gave an erroneous indication that the batch was okay. So, whenever you're doing sampling and whenever you're doing testing, always bear this in mind, that you could be committing an alpha error if you're rejecting a batch, or you could be committing a beta error if you're passing a batch. So, it becomes very important that your sampling plan is as robust as is practically feasible. Now, what is the practice in the industry? Formulators generally have a system by which, if it is an API, they do 100% sampling and check for identity individually and then composite everything and run the remaining tests. API manufacturers follow a system of root of n+1. Even the formulators for excipients follow the same root of n+1. Now, where did this root of n+1 come? WHO, for their own internal use, had come up with a sampling plan. At the very top, they mention in bold, this is purely for our internal purposes. Drug manufacturers are recommended not to follow these procedures, but to develop their own statistically valid sampling plans. But despite that, the industry has chosen to disregard this warning. Now, if you were to read the WHO sampling plan, they don't have just root of n plus 1. They have what is called the N plan, the P plan, and the R plan. In the N plan, it is where not too much information is known about the manufacturer. So they said over there, when you don't have too much information about the background of this particular manufacturer, you sample, if there are n drums, you take root of n plus 1. Now why they said root of n plus 1, I am unable to understand. Because if you ask a statistician, then he'll ask you, do you want a formal sampling plan or an informal sampling plan? And if you say it is an informal sampling plan, then he'll ask you the next question, is it a large population or is it a small population? Now if you ask him, what do you mean by a large population, what do you mean by a small population, then he might tell you that if the number of items in the population is more than 2,000, it is a large population. If the number of items is less than 100, then it would be deemed to be a small population, and in between it would be a moderate population. If it is a large population, he uses what is called the normal distribution. If it is a small population, he uses the gamma distribution. And if it is in between, he uses either the binomial distribution or the Poisson distribution or whatever. But in the absence of, you say, sometimes I have less than 100, sometimes I have more than 500, what do I do? Then he says, listen, as a thumb rule, root of n. Now, if it is 100, root of n is 10. But if it is an odd number and you get a fractional value, then what do you do? So he says, you can round it up, or you can round it down, or round it off. Now, that's exactly what the WHO people have said for their other plans. They have said that you can round up. That means if it is 11.4, then it becomes 12. Or you can round down. That means 11.4 becomes 11. Or you can round off. So 11.4 being less than 11.5 is rounded to 11. So these are the options that are available. So if you don't have too much information about a manufacturer's past track record, then please go for the N plan. So strictly speaking, it should be root of n, either rounded up or rounded down or rounded off, but on a consistent basis. Now, if you have some information about the manufacturer and it's a prestigious manufacturer, then they recommend the P plan. Now, what is the P plan? P plan says 0.4 times root of n. So if you have 100 drums, you don't take 10 drums or 11 drums, you take only 4 drums. Because the manufacturer is known to have a very good reputation. It's a prestigious manufacturer. But if the track record of this manufacturer has been erratic, he's a rogue manufacturer, then in that case the R plan. Now what does the R plan say? 1.5 times root of n. So if you have 100 drums, then you sample from 15 drums, not 10 or 11 drums. Now, there is no statistical power to this scheme, whether it is root of n or root of 0.4 times root of n or 1.5 root of n. The WHO sampling scheme does not have any inbuilt statistical validity. Now, if you want to run it properly, as all the regulators have kept telling you, you should have a proper sampling plan, a valid sampling plan. Then you need to refer to the various references that are available from the British standards, the US military standards, the Australia, New Zealand standard. Now here, when you are having these standards, they clearly bifurcate into two groups: sampling by variables and sampling by attributes. What is the difference? A variable is like weight, diameter, that you can measure, assay value that you can measure. In the case of attributes, it is qualitative, not quantitative. For example, your labels, they are smudged, or the color is slightly away from what you really require. It's almost there but not there. Now these kind of defects, these kind of differences are what we refer to as the defects by attributes. So the sampling scheme for attributes is different and the sampling scheme for variables is different. Now if you go to a statistician and tell him, listen, now I am told by the regulators that this root of n plus 1 may not be the best idea and that I need a statistically valid sampling plan, he'll ask you for 4 bits of information from your side. First, what is the specification limit? Now for purposes of our discussion, let us say the characteristic we are talking about is impurities. Specification limit that has been agreed between buyer and vendor for purposes of our discussion, let's say, is 1%. Now he asks you the next question. What is the AQL that you're looking for? Average quality level. Now, average quality level may not mean much by itself. So what it really translates to is the target level or the aspired quality level. So you have told your supplier that you are looking for something where the impurity level is not 1% but 0.3%. 0.3%. That's what you expect him to supply. That's what you are desiring. And then he asks you, what is the LTPD? The technical meaning of LTPD is lot tolerance percentage defective. Now by itself it may not mean much, but let me translate it as the lowest tolerable quality level. Now you don't want the supplier to reach the boundary, which is the 1% level. So you tell him that, listen, as soon as I see that your quality has deteriorated to 0.9%, I will start rejecting. So he asked you for what is the specification limit. You said 1%. What is the AQL? The average quality level or the aspired quality level or the targeted quality level, you said is 0.3%. Then he asks you what is the LTPD, the lot tolerance percentage defective or the lowest tolerable quality level, and you said that is 0.9%, not 1%, 0.9%. Now the last thing he wants from you is what is the alpha risk you're willing to take and what is the beta risk you're willing to take. In other words, how many times are you willing to risk rejecting a good batch? How many times are you willing to accept a bad batch? Because statistics is about uncertainty, dealing and coping with uncertainty. You don't have a black and white case. Statisticians don't have left-hand side equal to right-hand side. Left-hand side is always less than right-hand side or less than equal to right-hand side or greater than right-hand side or greater than or equal to right-hand side, but never ever equal to right-hand side. So there is an element of uncertainty. So you might say, fine, I will take a 5% risk for alpha, 5% risk for beta, or 10% risk for alpha, 10% risk for beta. Because the lower the risk level, the larger the number of samples that you will have to draw and test individually. Now once you have given him, let us say, specification limit is 1%, aspired quality level is 0.3%, the LTPD value is 0.9%, the alpha risk is 10%, beta risk is 10%, then he comes to you with what is called an operating characteristic in statistical language or quality control. It is known as the operating curve. It translates to something like this. N in brackets C, comma, C plus 1. What does it mean? N is the total number of samples that you have to individually draw. C is the total number of out of specification readings that you get, but which is acceptable. And C plus 1, the moment it crosses that level, then you reject that batch. Let me explain it in a little more simple way. He gives you a figure called 24 in brackets 4, 5. What it means is, please draw 24 samples, run those samples individually, don't composite them. Run them individually. Up to 4 out of specification readings, ignore it and pass the batch. But the moment you get the 5th, you have to reject the batch. Now this operating curve, the shape of the curve has much to say about the robustness of the sampling plan. It has a y-intercept and it has an x-intercept, and in between you have the curve. The part near the y-intercept is usually referred to as the head of the curve. The portion near the x-intercept is referred to as the tail of the curve, and in between is the body of the curve. If the slope of the curve at the y-intercept is steep down, it indicates that there could be a high amount of alpha risk or alpha error. However, if the head is flat, from the y-intercept for a portion, the curve remains parallel to the x-axis more or less, and then comes down, it shows that the alpha error is very low. It comes as close as possible to an ideal sampling curve. Similarly, the body of the curve, if it is closer to the y-axis, the more close it is to the y-axis, the lower the beta risk or the beta error. For example, in an ideal operating curve, you would have a flat head and then a steep down parallel to the y-axis, and that would mean 100% robustness. But that is not possible in statistics. You have an alpha error, you have a beta error, you have to live with it. So if the curve is such that it is flatter towards the y-intercept and then comes down sharply and stays close to the y-axis, then the beta error also is reasonably low and the sampling plan is robust. I'll give an example here. You have 3 different operating characteristics. It says n equals 200, that means you draw a sample of 200. C is equal to 4, that means up to 4 non-conforming units are permitted. The next curve is slightly away from the y-axis, and it says draw 100 samples and up to 2 non-conforming units are permitted. And lastly is the curve which is furthest away from the y-axis, which says sample 50, up to 1 non-conforming unit is permitted. So as you can see, the beta error increases as the sample size decreases. Now how could you use this? As you have, for example, in content uniformity, what is called multi-layer testing, multi-level testing. You take 10 tablets and assay them. If all of them pass, you pass. But if one of them is outside 85% to 115%, but within 75% to 125%, then you take 20 more and assay them. If all of them are within 85 to 115, pass the batch. Otherwise, fail the batch. The RSD in the first case, if you have taken 10 tablets, should be less than 6%. And when you have taken another 20 tablets, the total RSD can go up to 7.8%. I'm just giving you an example of one particular test where you have multi-level. For example, similarly for dissolution, you have S1, S2, S3 or L1, L2, L3, where you take 6, and then if there is one problem there, you take 6 more, and if there's yet one more problem there, you take 12 more, and when all those 24 together are evaluated and it is all right, you release the batch. So similarly, over here, the statistician may tell you 24 in brackets 4, 5. So what does it really mean? It could mean, and he is the best person to explain it, it could mean 12 in brackets 2, 3, 6 in brackets 1, 2, and 3 in brackets 0, 1. What does this mean? He says you take out 24 samples, but you run only 3 at random, you pick it up. And if all 3 are within specifications, pass the batch. But if there is one which is out, you take 3 more from the sample that you've already drawn. And if all those are all right, you pass the batch. Because out of 6, you have only 1 out of specification, all the remaining are all right, you pass the batch. But if there too you get 1, which is 1 more, that means you have now 2 out of specifications, then you take 6 more and assay them. And if everything is all right, then you pass it, because the sampling table allows you 12 in brackets 2, 3. So you have had only 2 out of specification out of 12 assays, so you pass the batch. But assuming now you get one more, you take the remaining 12 and assay them. And after 24 tests, you have less than 4 out of specifications, you can pass the test. It's all from the same distribution, so it's all right, you can pass the test. But if you have 5 total out of specifications, then this batch certainly does not meet your requirements. Now, how do you get those 24? You don't go there and just pick it up. No, you should have what is called a random number generation facility. Now, any statistician will help you with that, but I'll try to make it simple and explain it to you. See, very many people, when they draw from drums, they use a thief which has got 3 compartments. There's one at the bottom, one at the middle, and one at the top. So they really are sampling 3 separate samples. But what do? They composite it. Then the whole purpose is lost. Now, if you look at a drum, and let's say it's a circular drum, the part away from you is north, the part towards you is south, the part to the left is west, the part to the right is east. So you have N, S, W, E. Then you have the top T, middle M, and bottom B. Now, if you have, let us say, 100 drums, and he tells you, you take 24 samples. So how do you get those 24? You have a small program which generates random numbers, and you tell it to generate, you say the total number of drums is 100, the number you wish to sample is 24. Now please tell me how to draw those samples. So it will tell you, go there and number all the drums as 1, 2, 3, 4, 5, 6, 7, 8 till 100. And then it tells you 7 NB, 11 MW, and so on and so forth. What does that 7 NB means? The 7th drum that you have numbered, the northern part that is away from you, B, the bottom, pick that. Then go to the 11th drum, and from the western side, you draw from the middle, and so on and so forth. You get the total number of 24. Now, from this 24, you ask one more time, I need to take 3. Which 3 should I take? The random number will tell you 26 E M, 52 T S, and maybe 76 W B. So now, if those are there, those are the numbers that are already with you. You have entered that into the computer, and from that you have asked the random number to select 3 for you. It will tell you those 3. Now you run those 3. And once you've done those 3 and all those assays pass, and within specification, pass the whole lot. This is called statistically valid sampling. Planned and execution of a statistically valid sampling process and testing. There is randomness, there is total independence. Every drum had an equal chance of being sampled. Every location in every drum had an equal chance to be sampled. We have talked about vendor qualification, where you have a vendor status which is approved, preferred, and most preferred. So when you sample, you always sample 1.5 times root of n. Always. But when he is in the approved state, you run all those that you sampled. Once he goes to the preferred, you go to the n plan. So you do only root of n. Although you have 1.5 root of n sampled and ready, you test only root of n. And if he goes into the most preferred category, then you have sampled 1.5 root of n, but you run tests at random from this sample of only 0.4 root of n. So not only do you only do the reduced testing in terms of the first 3 batches, you ran all tests, and the next 3 batches you did 50% of the tests, and then after that, when he's in the most preferred category, you do only the identity test and 1 or 2 critical tests. Now the number of samples too, you choose only 0.4 root of n and run that identity test and 1 or 2 other characteristics. Assuming you have a problem, then you take additional samples from here that you already sampled. You're not doing fresh sampling. It's already sampled and kept for you. So you took out 4 and ran the assays or the tests, and you have a problem there. Then you take root of n and whatever is the additional amount. You have taken only 0.4 times root of n. Now you take the remaining 0.6 root of n and run those tests. And if they pass, you release the batch. But if there is a problem there too, then you sample all the 1.5 root n that you have sampled and run the test. And if it passes, you pass. Otherwise, you have a problem and there is rejection. And this arrangement is already discussed with and agreed by between buyer and vendor. So I repeat, once a vendor is in the most preferred category, you have 1.5 root n samples drawn and kept ready, but you run only 0.4 root of n tests, and that only for identity and 1 or 2 characteristics. If there is a problem there, if there is no problem there, you pass it. But if there is a problem there, you take additional 0.6 root of n that is still lying here and run those tests. And if you have no further problems, you release the batch. But if you still have a problem, you run all the 1.5 root of n that you have sampled and see if it passes. If it passes, you release it. Otherwise, you reject it. So this is one way of linking the samples that you test, the number of samples that you test, to a vendor's status. So vendor status grading helps you justify reduced testing, and sampling is done at the same level, but the testing is only from that many of those that are already sampled and kept, and the rest of it remains with you as reference samples. It has been a general practice in the pharmaceutical industry to have the QC lab located far away from the production space, for fear that the production environment may interfere with the tests that are carried out in the lab. But now the regulators have taken a different view, observing the practices in other industries doing similar work, in the chemical industries, for example. Now they would recommend better and better instrumentation for obtaining as much as possible of real-time data. So they have now 4 different types of testing. Offline testing, which is the same as in the past. You take a sample and go far away to a lab located at a remote location. Then they have something called at-line testing they recommend. Make an environment very close to the process which is suitable for testing, and draw samples and test it as close to the process as possible and release the results so that the process may be improved upon or kept in a state of control. The next thing they talk about is online testing. Online testing refers to drawing a sample stream from the process and returning the stream to the process and using the probes on that sample stream. And finally, the last type of testing they are advocating is inline testing, where you put the probe directly inside the process and monitor the data. So you have now 4 types: offline testing, at-line testing, online testing, and inline testing. What the regulators are trying to promote is that you try and get as much real-time data as possible so that your process is always maintained in a state of control, so that you don't discover at an awkward time after the process is completed that things have not gone quite the right way. Now, in case of in-process sampling, SOPs should be very clear. If it's a liquid, are you using tip tubes? If it is solids, are you using a falling stream? If you're using a sampling implement, how do you clean it? How do you approve it for use? And how do you approve it as